Great. Now I have two microphones. So thank you. We're trying to stay on time. If you could please keep your questions until the end of the session, that would be great. So our next speaker is Dr. Maguire, who is a professor in the Department of Ophthalmology at the University of Pennsylvania School of Medicine, where he is a retina specialist and a vitreoretinal surgeon. Dr. Maguire is most well known for developing and carrying out the surgical procedures that are now used in a large number of gene therapy clinical trials using interventions for congenital blindness, and he will tell you more about it.
So today I will discuss the development of a novel outcome measure which was critical for the approval of gene therapy for a form of congenital blindness. Just to orient you to our project, Leber congenital amaurosis is caused by a lack of function mutations in the RPE65 gene. This gene encodes an enzyme which converts vitamin A into its active form necessary for vision. Affected animals and humans have early onset blindness, abnormal eye movements, and flat electrical responses to light stimulation. I would also argue, in addition to night blindness, they have day blindness. In a room like this, where we perceive this amount of light, I would have to remove every light bulb except one and spread that light across the entire area of the ceiling for you to get the sense of how day blind they are. So using gene therapy with an AAV carrying a wild-type RPE65 gene, we were able to restore some vision to affected individuals. And for our phase 1 trial, we chose an outcome measure, pupillometry, which provided objective evidence for improved function. Pupil constriction was much stronger to exposure to the treated eye, and when the other eye had a light flash, the pupils dilated, a Marcus gun or afferent pupillary defect. This objective test seemed to me the formula to follow for the FDA phase 3 trial or efficacy trial, and I would be absolutely wrong. The FDA, by federal mandate, requires that for a drug to be approved, there must be a demonstration of clinically meaningful improvement. Pupillometry was not considered a clinically meaningful outcome. As one reviewer put it, patients care about vision, they don't care about their pupils, and that's absolutely true. So at the time, there was no recognized outcome measure considered clinically meaningful except the surrogate 3 lines of improvement on an eye chart. And while some patients showed an improvement in visual acuity, which is central vision, the main improvement with this drug is rod photoreceptor mediated, which is side vision and night vision. And at the time, there was no test of this type of vision that satisfied the clinically meaningful, clinically significant mandate. In the phase 1-2 trial, we had looked at mobility testing, essentially an obstacle course, as an exploratory endpoint. And this is an example of one of our earlier versions. So this is a pre-injection on the left and 3 months post-injection. It takes that child 6 minutes to go through the obstacle course on the left versus a few seconds on the right. The FDA suggested that this smelled clinically meaningful, so we were essentially assigned the task of creating an entirely new outcome measure based on this framework. The FDA provided some excellent feedback on developing this into a standardized, statistically rigorous test that would satisfy the clinically meaningful mandate. We were now primarily interested in functional vision, that is, vision that impacts activities of daily living as opposed to visual function. The multi-luminance mobility test is a novel metric that measures the speed and accuracy with a subject can ambulate independently under different ambient light conditions. The MLMT is essentially an obstacle course that measures a subject's ambulatory vision in different environments. So this is a bird's eye view of one layout of the course. There are 12 layouts total, and the choice of layout is randomized for each run. The test is done at light levels ranging from 1 lux to 400 lux, which corresponds to various light conditions encountered in daily living. For example, 1 lux is a moonless summer night, 400 lux is a bright office building, and 50 lux a outdoor train station at night. So subjects are dark adapted for 40 minutes and then asked to navigate the course as quickly as possible with the fewest errors possible. At baseline, the lowest light level that a subject can achieve a passing score is identified, and this is used to bracket the subsequent testing conditions, and each run is video recorded. So the MLMT was developed with input from the FDA to have both rigor and objectivity. So on this side, the light levels in the room are standardized and calibrated. Exposure of the videos is adjusted before they are sent to the graders, so the graders cannot identify the light levels of the videos that are used during the test. Each of the 12 layouts has the same number of turns and obstacles. Subjects experience a separate practice course to mitigate the potential of a learning effect. Examiners in attendance do not provide guidance to the subject, and that is monitored on audio as part of the grading procedure. So graders are masked to the treatment status. They receive videos in a random sequence such that they might encounter a video of a test done post-treatment before grading a test done pre-treatment. And each video is graded by 2 graders and adjudicated by a third if necessary. And as a quality measure, the videos are randomly selected for repeat grading. Each run of the MLMT is scored and graded for accuracy and time. Patients must pass both the accuracy and the time components to earn a passing final score. For example, if a subject is unable to finish the course in under 3 minutes, that's an automatic fail. Each light level is assigned a score code. 1 lux is 6 points, 4 lux equals 5 points, and so on. And from this score code, a change score can be calculated over time to measure improvement in ability to navigate, and nonparametric statistical tests are applied. So a validation type study was done initially in normal-sighted control subjects and patients with inherited retinal diseases to measure construct and content validity. Subjects were tested twice at study entry and again at 1 year. All 25 normally sighted subjects passed at the lowest light level and showed no difference in change score over a year, whereas none of the 28 inherited retinal disease subjects improved over 1 year, and 8 of them actually got worse. So the MLMT was easily able to distinguish between normally sighted and visually impaired subjects. And on the left here, you see a tight cluster for the normally sighted patients in terms of time and accuracy, and all passed. And on the right, the visually impaired subjects exhibit a range of performance as we expected. So looking at the affected patients, we looked at the relationship of visual field and MLMT performance. And on the left, the graph shows that subjects with smaller visual fields, so this is about 20 degrees of field. If you had a smaller field, you were much more likely to fail as opposed to patients with a bigger field. And this correlates just a little bit bigger than the visual field that defines legal blindness. A similar trend was seen with visual acuity. And again, here, LogMAR acuity, when it's greater, means worse vision. And again, as the vision is worse, there are more failures. We also looked at intra-grader and inter-grader agreement. And for the major component of overall pass-fail status and for all subcomponents, agreement was typically above 95% with an N of over 4,000 videos. So looking at the results of the phase 3 trial, we can see that the MLMT provided clear evidence for a statistically significant improvement in visual function for patients receiving gene therapy. In addition, the results of the MLMT correlated nicely with measures of visual function such as sensitivity, which is the ability to perceive different ambient light levels, and for visual field, both Goldmann visual field and central vision field, Humphrey visual field. Looking more closely at the mobility results, we also see, not surprisingly, that treated patients were able to move through the course much more rapidly. So here we see that at the same ambient light level, they were able to go through a course 52 seconds faster, whereas there's no significant change in the control. Moving through faster correlates to the real world, like walking around Washington, D.C. and avoiding traffic. So, frankly, though, all the graphs and statistics aren't nearly as compelling as the raw data itself. The improvement is immediately obvious. We see here on the left pre-treatment and on the right 1 year later. This is not a visual acuity test. People are often improving without any change in eye chart acuity. It somehow incorporates light sensitivity, visual field, and other parameters of visual function. So, to summarize, the MLMT is a novel test that was developed to provide a primary outcome measure for subjects receiving investigational products for inherited retinal dystrophies resulting in reduced retinal sensitivity and visual field. And its essential features include the ability to differentiate low vision from normal subjects, detect changes in performance over time, identify a wide range of performance among the visually impaired. It's highly reproducible. We have demonstrated construct and content validity, and most importantly, it provides a quantifiable measure of a clinically meaningful functional vision. Thank you.
Thank you very much, Dr. Maguire.